Hello, my name is Joanna Sajewicz-Kutkowska. I work in the Department of Poultry Diseases at the National Veterinary Research Institute in Puławy, Poland. And what I'm going to do today is presenting the transcriptome analysis of the spleen following chicken astrovirus infection. Astroviruses cause enteritis in humans and various animal species. In poultry, they cause enteritis combined with growth depression and higher mortality, but their presence was also noted in healthy flocks. Chicken astrovirus was recently indicated as the white chick syndrome factor. White chick syndrome is associated not only with increased embryo or chick mortality, but also with weakness and white plumage of hatched chicks. Astroviral infections pose significant problem in poultry industry, leading to decreased egg production, breeding disorders, poor weight gain, and even increased mortality. Chicken astrovirus-mediated pathogenesis in chicken occurs due to complex interactions between pathogen and immune system. And these interactions, virus host, remain unclear. There is also no information about global gene expression changes in chickens in response to chicken astrovirus. That's why the aim of our study was to investigate the molecular background triggered by chicken astrovirus infection. All the animal experiments were performed in accordance with the requirements of the Ethical Commission. Chicken astrovirus strain from 2014 was propagated on embryonated SPF chicken eggs. We had two experimental groups, and the first one infected with chicken astrovirus, and the second one, the negative control, inoculated with PBS. We had 5 birds per group. The inoculation was performed per os using 200 microliter of isolate, and the birds were housed in HEPA-filtered isolators for 4 days. And at 4 dpi, the animals were euthanized, and we collected the spleen samples. These samples were homogenized. Then we extracted the total RNA using commercial kit from Qiagen. We measured the concentration of isolated RNA with qubit. And we also checked the RNA integration number using bioanalyzer, and we had this threshold of 7 for all of the samples. The cDNA libraries were generated using TruSeq-stranded mRNA-LD sample prep kit from Illumina, and the sequencing was performed at the external service, Macrogen Europe, on Novasec from Illumina. At the end, we performed bioinformatic analysis, and shortly we used FastQC for quality control, Trimomatic for trimming, StarMapper for mapping to the reference, StringTie for merging, and Balgon for expression calculation. We also performed differentially expressed genes analysis in the SEQ using bioconductor, and functional analysis with gProfiler based on gene ontology and KEGG databases. And the obtained results were validated with qRT-PCR. The global profile of gene expression in the chicken spleen is a good overview of the host response to chicken astrovirus infection. We chose spleen, the secondary lymphoid organ, because it effectively induces innate and adaptive immune responses, which are very important for birds because of their underdeveloped lymphatic vessels and lymph nodes. Infected chickens had no symptoms up to 4 DPI. We also did not observe any visible differences in the spleens between control and infection groups. But the chicken astrovirus replicated in the chickens because we detected the viral RNA in clagal swabs at 4 DPI. We identified almost 32,000 genes in response to chicken astrovirus infection. When we compared the infected and normal group, we identified 204 significantly differentially expressed genes, with 152 upregulated and 52 downregulated. We then filtered these results with the threshold of p less than 0.05 and log-to-fold change bigger than 1, and we identified 45 DIGs with 26 upregulated and 19 downregulated genes. As I said before, we validated these results with QRT-PCR method, and this validation was performed for 4 genes, and the QRT-PCR results confirmed the transcriptomic results. On the slide, you can see 2 figures. The first one is a heat map analysis used to classify gene expression patterns under different experimental conditions. And the second one is a volcano plot, which displays the number of deaths between the control and chicken astrovirus infected groups. To identify DEC functions, we performed a gene ontology analysis, and we were able to annotate the genes into 3 groups: biological processes, molecular functions, and cellular components. Among biological processes, the most enriched processes were these related to cell cycle, immune response, and regulation of biological processes. Among molecular functions, the most enriched processes were related with binding activity, including ATP binding. And among cellular components, the most enriched categories were relevant to intracellular components. The most upregulated genes were mainly involved in immunity and defense, and also in response to and regulation of viral or biological processes. While the most downregulated genes were mainly related to the regulation of biological processes. We used KEGG database to analyze pathways and to define DEC functions. We found the enrichment of genes associated with immune-related pathways like node-like receptors signaling pathway and influenza A pathway and associated with cell cycle. RNA-seq generates large amounts of data. That's why today I will focus only on our key findings. And the first one is a cell cycle arrest promotion, which was accompanied with the inhibition of genes encoding T, MEM158 and YWHAB, and with the increase in the expression of CDKN2C. We have no examples, no information in the literature about the role of MEM158 in viral infections. But we do know that the silencing of this gene in ovarian cancer cells promoted G1 phase arrest. We also know that 14-free protein family participate in control of the cell cycle in metabolism, apoptosis, and gene transcription. 14-free protein interacts with the HIF protein and regulates the cell cycle by inducing G2M arrest. And the last one, CDKN2C, encodes an inhibitor that prevents activation of the CDKs and controls the G1 phase of the cell cycle. These CDKs are the key regulators of transitions from one phase of the cell cycle to the next one. Our functional analysis also revealed that the JUN gene was downregulated in both of the enriched pathways associated with the antiviral response. I mean the node-like receptor signaling pathway and influenza A pathway. This gene takes part in the development of viral infections and can be activated by extracellular factors, including viruses. We know that suppressing of JUN in human and mouse cells efficiently reduces avian influenza virus replication. So the suppression of JUN gene expression in chicken astrovirus infection may be the evidence of the immunosuppressive effect of chicken astrovirus. Another example of the immunosuppressive nature of chicken astrovirus appears to be its effect on the complement pathway. We showed that C1R and C1s complement pathway genes were upregulated, while we showed the lack of activation of other components of this system, which means that there is the inhibition of this pathway at this stage. We need to remember that the crucial role of the complement is to start inflammatory response. So the absence of inflammation in astrovirus infection suggests suppression of complement activation by astroviruses. So we have demonstrated that chicken astrovirus infection in chickens affects both the cell cycle and immune signaling in the spleen. Most overexpressed DEGs, mainly interferon stimulated genes, are putative interferon-induced chicken astrovirus restriction factors. And most of the downregulated genes are associated with the cell cycle and stress response. We hypothesize that chicken astrovirus have a mechanism of immune evasion that prevents the significant activation of immune genes in the early stages of infection, but this may also depend on the virulence of the virus and the age of the chickens. But in conclusion, although our study had some limitation, like the small study groups and single time points, it provides a global analysis of host transcriptional changes that occur during chicken astrovirus infection in vivo, and a strong basis for further studies. Thank you for your attention.